Bom, boa tarde a todos e todas. É com imenso prazer e satisfação que a gente dá início a essa oficina virtual em capacitação para a defesa da saúde pública, apropriação de custos de produção, química fina e impacto das patentes farmacêuticas, com Joseph Fortunak e Eloan Pinheiro. Agradeço imensamente, em nome do GTPI e da ABIA, a participação do professor Fortunak e da Eloan. É uma honra tê-los aqui conosco. Essa oficina, ela terá tradução simultânea para português e inglês. Então, aqueles que precisarem, é só ir aí no Zoom e habilitar a interpretação. Bom, essa iniciativa, ela busca fornecer conhecimento e ferramentas essenciais para a gente compreender o complexo cenário que envolve a produção de medicamentos, a propriedade intelectual e seus impactos diretos na saúde pública. A gente tem como objetivo capacitar ativistas, profissionais de saúde, químicos, economistas e todas as pessoas interessadas em defender os direitos humanos, a vida e uma política pública soberana que promova o interesse público. A gente espera, a partir desse curso, termos novos parceiros e projetos para uma verdadeira regulação social dos preços de medicamentos e para defender o nosso Sistema Único de Saúde. Eu começo apresentando a Eloan Pinheiro. Eloan é uma renomada profissional, tem uma trajetória notável na área da química e da saúde pública. Graduada em Química em 1972, com especialização em tecnologia farmacêutica pela Universidade de Londres em 1990. Ela iniciou sua jornada profissional em 73, trabalhando nas indústrias farmacêuticas Beckham e Sidney Ross, com formulação de medicamentos e síntese química. Em 1988, Eloan passou a atuar como consultora da rede de laboratórios oficiais. Sua dedicação à saúde pública conduziu para a Fundação Oswaldo Cruz, onde, em 1994, assumiu o cargo de diretora do Instituto de Tecnologia em Fármacos, uma posição que ocupou até 2002. Durante sua gestão, teve um impacto significativo na pesquisa e desenvolvimento de medicamentos antirretrovirais. Eloan se aposentou em 2004, mas como a gente pode ver, ela segue trabalhando com imensa paixão, defendendo a saúde pública e causas humanitárias. Ela contribuiu com seu conhecimento e experiência internacionalmente, trabalhando no Departamento de AIDS da Organização Mundial da Saúde de 2004 a 2006. Além disso, Eloan se dedicou a projetos de saúde global como consultora da Unitaid de 2007 a 2013. Atualmente, Eloan continua sua atuação como consultora em saúde pública em escala global. Sua paixão, dedicação e experiência fazem dela uma figura inspiradora, respeitada entre ativistas, pesquisadores e gestores da saúde. Professor Joseph Fortunak é professor titular nas áreas de Química e Ciências Farmacêuticas na Universidade de Howard. Além disso, ele é diretor científico da empresa de cannabis, Cannabidiol. Fortunak possui uma vasta experiência com centenas de publicações em revistas acadêmicas, revisadas por pares. Tem também patentes e apresentações em simpósios científicos. Ele é um renomado educador, lecionando sobre a produção de medicamentos e a garantia de qualidade da fabricação, regulamentação farmacêuticas em múltiplos locais na África. Sua paixão pela promoção do acesso responsável a medicamentos a preços sustentáveis o levou a colaborar com diversas empresas e organizações nesse esforço. Fortunak desempenhou um papel fundamental no lançamento de mais de 40 produtos inovadores e genéricos. De 1983 a 2004, ele trabalhou na indústria farmacêutica, desempenhando funções que variaram entre cientista de laboratório, inventando os novos produtos e processos químicos, até gerente com supervisão de até 500 profissionais. Participou também do lançamento de 15 novos medicamentos e 25 novos produtos genéricos, que desempenharam um papel crucial em áreas terapêuticas como AIDS, doenças cardiovasculares e sistema nervoso central. Colaborou com iniciativas como o Global Fund, Unitaid, Médicos Sem Fronteiras e o Medicines Patent Pool. Também trabalhou com diversas empresas de genéricos, com destaque na Índia e na China. Bom, como vocês podem ver, o currículo desses dois participantes é imenso e estamos aqui diante de duas figuras que vão nos auxiliar bastante. Eu desejo a todos uma excelente e proveitosa oficina e seguiremos em defesa da saúde pública. Passo a palavra já para a Eloan.
Boa tarde a todos. Bem, é um prazer enorme estar aqui, junto com esse meu grande amigo e querido amigo, Joseph, no qual a nossa parceria data de longa, de anos atrás, tentando trabalhar e esclarecer esse mundo que é esse mundo farmacêutico, esse mundo que é esse mundo de medicamentos, esse mundo que é esse mundo de preços exorbitantes e pouco acesso àquelas populações mais necessitadas. Eu inicialmente vou começar a minha apresentação e depois vou passar para o Joseph, porque a sequência que a gente escolheu foi, inicialmente, eu vou começar, depois vou parar, e Joseph, então, entra com a parte fundamental dos IFAs, que são os APIs, e amanhã eu volto com a apropriação de produtos formulados. Então, com isto, a gente vai dar uma ampla visão a vocês que são militantes, a vocês que lutam por uma saúde pública consequente, de acesso a medicamentos e contra a exclusão social que hoje os grandes monopólios fazem para ter uma saúde mais uma qualidade de vida melhor. Eu acredito que com isso a gente possa contribuir para vocês levarem as bandeiras de luta junto a seus governos de uma forma bem concreta e consequente. Como eu estou com dificuldade de entrar com meus slides, eu vou pedir a Suzana para me auxiliar, porque é a primeira vez que eu tenho que fazer isto, entende? Até amanhã eu treino melhor. Ok. Suzana, página inteira? Ok. Bem, o nosso curso de apropriação do custo dos antirretrovirais. Por que é importante precificar medicamentos? Quais são os principais problemas que afetam o acesso aos medicamentos? E como superar os entraves ao acesso dos medicamentos? O que a gente tem visto hoje em dia é cada vez mais um menor acesso das pessoas, das populações, inclusive aquelas mais pobres, aos medicamentos que elas necessitam. Seja porque a pesquisa não entra naqueles medicamentos das doenças negligenciadas, seja porque a gente tem um sistema hoje que eu considero extremamente cruel, que eu chamo de patentes de produtos. Pode passar, Suzana. O que é de fato uma patente? É uma concessão que quem dá é o Estado, e vocês, como militantes, conhecem isso mais próximo do que eu. O Estado concede a ao setor privado, a uma empresa, o direito de explorar o objeto que eles descobriram ou uma descoberta. Ele permite que ele comercialize de forma exclusiva, monopólica, esta patente. Porém, uma patente, normalmente, ela tem que ter novidade absoluta, atividade inventiva e aplicação industrial. Isto é a patente, aquela que tem aplicação industrial, senão o nome fica sendo propriedade intelectual. As empresas farmacêuticas afirmam que a patente é uma legítima recompensa para os elevados níveis de investimento e o risco associado ao desenvolvimento de novas drogas. E que essa recompensa legítima é o que o Estado lhe fornece como monopólio. Esse direito de comercializar, de fixar o preço, de impedir terceiros a fazer esse mesmo produto pelo prazo de 20 anos. A próxima. A próxima, Suzana. Ah, para mim, assim, é tão interessante. A gente volta, Suzana, por favor, a anterior. Há tempos atrás, essa minha luta contra a patente começou na década de 90, porque foi na década de 90 que a instituição de patentes ela foi mais fortalecida, porque ela sempre existiu, ela existe há muitos anos, somente com diferenças ao longo do tempo. Antigamente, você não patenteava produto, que era o API, o IFA. Antigamente, você tinha somente patente de processo. Em alguns países, inclusive na década de 80, eles não reconheciam patentes para medicamentos. E, com isso, houve um grande avanço na indústria farmacêutica. Mas o que está escrito aqui é uma coisa feita pelo Oxfam em 2000, se não me falha a memória, em 2003, isto é, 20 anos atrás. Que ela diz o seguinte: como o pobre pode comprar remédio se ele não pode alimentar as suas crianças? O rico pode pagar tratamento médico e comprar remédios. Para o pobre, os remédios são um luxo demasiado para a sua imaginação. Isso foi dito por um trabalhador em Zâmbia. Um outro afirmativa foi de um trabalhador rural vietnamita. Para a população pobre, a doença é uma constante ameaça. É uma tarefa muito difícil para nós a compra dos remédios que necessitamos para tratar nossas crianças, até mesmo quando elas têm uma simples febre. E também foi dito pelos Médicos Sem Fronteiras: os medicamentos inovadores estão ficando nos países em desenvolvimento cada vez mais inacessíveis para aqueles que não fazem parte de uma elite privilegiada. O impedimento de produção local simplesmente excluirá a maioria da população do acesso aos remédios. Por outro lado, o que a indústria fala? Naquela época falava, isso foi ano 2000, ano 2000 mesmo. Nossa força conjunta nos capacita a estabelecer uma rede globalizada, setor privado, governo, que forma a base daquilo que se transformou no TRIPS. Isso foi o executivo da Pfizer. Um relatório anual da Glaxwell, como diz, o enfraquecimento dos direitos da propriedade intelectual, só serve para reduzir os incentivos para a pesquisa de novos remédios, incluindo a inovação para doenças negligenciadas. Grande mentira, porque se você olhar, você pouco tem de remédio de doenças negligenciadas feito por essas grandes empresas. Os direitos de patente estão se tornando cada vez mais complicados e abrangentes, e os ganhadores no sistema de propriedade globalizada serão aqueles que tiverem recurso para entrar no jogo. É muito difícil imaginar como os países mais pobres do mundo possam estar entre os ganhadores. O desnível é muito grande e ele representa um risco real de falhar em se alcançar um futuro melhor. Essa Philippa Saunders, Projeto de Medicamentos Essenciais da Grã-Bretanha. Portanto, essa discussão é muito antiga. Eu sempre digo assim, eu estou desde a década de 90 discutindo que patente é um mal para a sociedade, um mal para a população carente, um mal para a população que não, de baixa renda. Pode passar o próximo. Patente se tornou, para mim, uma arma sem pólvora. pois pode aniquilar globalmente aqueles menos favorecidos. Hoje temos patentes para vacinas. Covid matou muitos que poderiam ter sobrevivido. O lucro da Pfizer em 2022 foi de 100 bilhões de dólares e nós tivemos 6 milhões de mortos de AIDS. De AIDS, não, só, desculpe, de Covid. No Brasil, 700 mil. Patente para medicamento tuberculose. Tuberculose é uma doença negligenciada. A bedaquiline, patente para medicamento que trata AIDS. Vários produtos são patenteados. Vários produtos, patentes para hepatite. Vários produtos para câncer, todos sob patente, que faz com que os preços cheguem, são estratosféricos e as pessoas morrem por não terem acesso e os governos pobres não têm orçamento para cobrir a demanda social que existe em seus países. As formas de patentes também hoje são várias. Patente para combinação de drogas. Hoje você tem 2 drogas, uma já caiu patente, a outra está sob patente, eles combinam e fazem uma nova patente. Isso é que é um controle econômico internacional do comércio de medicamentos. Eles patenteiam para drogas, como o tenofovir, o TAF. Eles patenteiam, está entendendo, sem nenhum critério, o mitiu, como ela lamivudina e tricitabina. Eles patenteiam, antes era a lamivudina, aí caiu a patente, aí eles patentearam a tricitabina. E a gente, vocês que são, que fazem advocacy em relação à patente, e quando a gente vê uma patente, a gente vê as vergonhosas patentes Marcus, aquilo que ele pega o núcleo farmacofórico e vai juntando um monte de, agregando um monte de componente e patenteiam toda uma família. Isso Fortunak vai falar muito mais, com muito mais propriedade do que eu, que estou longe da química fina há muito tempo. Mas estou próximo da saúde pública, estou próximo das formulações. O próximo. Consequências é essa aí. Essa pergunta, essa charge, como é que é? Um remédio que faz emagrecer, abaixa o mau colesterol, aumenta o bom e pode comer de quase tudo. O que mais ele faz? Aí a empresa responde, me dá $1 milhão de reais ou dólares por mês. A outra são imagens realmente concretas. Patentes na Organização Mundial do Comércio, é isso aí. Igual ou pior que as patentes são os trade secrets, quer dizer, a gente tem tudo na OMC, a gente tem todos os tipos de trade secrets, contrato, patente. As indústrias é muito engraçado, elas consideram importante o livre mercado, mas patentes, trade secrets, impede a livre concorrência, quer dizer, é uma coisa interessante quando você tem determinados objetos econômicos, como petróleo e outros, que eles defendem, não, o petróleo não pode ser o monopólio, etc., eles têm que ter um mercado concorrencial, né? Quando você fala o mercado concorrencial para outros insumos que não farmacêuticos, mas quando você fala do mercado de patentes, né, de farmacêutico, eles defendem o monopólio, eles defendem absoluta os trade secrets. Os trade secrets são aqueles contratos que eles não têm nem tempo, nem término, nem prazo para término. Eles são absolutamente fechados e não acessíveis. Por favor, outro. Patentes garante lucros e mata pessoas. Bem, patente, vocês sabem muito bem, colocada da TRIPS na OMC em 1995. A OMC foi criada e a TRIPS foi colocada lá. Isso foi durante a pandemia de AIDS, e isso criou uma situação extremamente complicada no mundo. Mas, evidentemente, fizeram algumas salvaguardas. dizendo, né, fez a reunião de Doha e as salvaguardas que vieram, que eu não considero de jeito nenhum salvaguardas, e vou dizer por quê, né? Porque a licença voluntária, que é tão defendida por muitos, né? E é considerada também uma das salvaguardas assumidas pela unidade, ela mantém uma relação de poder das empresas farmacêuticas. O poder está ali. Ela concede licença voluntária a um outro fabricante, mas, ao mesmo tempo, ela define as regras geopolíticas de mercado. Ele vai licenciar aonde ele quiser, a quem quiser e fazer o preço que quiser. Tem a quem atender contrato com empresas públicas ou privadas não transparentes, tempo de contrato, royalties, pagamento de know-how, mantém sob controle o mercado de intermediários, acredito que o José vai falar muito bem sobre isso. Às vezes você tem também, dentro desse controle dessas licenças voluntárias, o controle sobre o mercado do intermediário que chegaria a fazer o fármaco. E mantém a comercialização em contratos fechados, mesmo após o término da patente. Quando eles fecham esse contrato, às vezes os patentes se extinguem no meio do contrato, mas aquele contrato não se extingue, que é um absurdo, que é uma coisa absolutamente ilógica. Eu quis fazer essa introdução para vocês de patente, que vocês são atuantes em patente. Eu nem sou uma pessoa que atua em patente, eu atuo no acesso ao medicamento, eu atuo na questão de apropriação de custos de medicamentos, eu atuo na questão de direitos humanos. Mas vocês conhecem e vocês sabem, com muito mais detalhes do que eu, as complexidades que são a questão desses contratos de licença voluntária. Próximo. Próximo. Como enfrentar esta situação? Para mim, só tem uma saída, é licença compulsória. Para mim, o momento atual tem que ter um momento de luta direta contra esses altos preços que existem. Porque reproduzir uma patente necessita de tempo. Porque, por mais bem detalhada que seja uma patente, os detalhes do processo produtivo não estão descritos, tais como os parâmetros termodinâmicos. Quando eu trabalhava na Beecham, eu ainda trabalhava em síntese, havia a síntese da amoxicilina. Nós usávamos uma série de detalhes no processo industrial que não estavam na patente. Por exemplo, a gente usava na adição de reagentes um tempo muito específico, não descrito na patente, que garantia melhor pureza e maior rendimento do produto final. Era, assim, uma maneira, esses truques, e eu acho que o José vai falar bem sobre isso, essas coisas, esses dados termodinâmicos, velocidade reacional, tempo de adição dos reagentes, temperatura, não está na patente descrito esse lead time, não tem isto na patente, eles colocam sempre um range, quer dizer, uma faixa. E isto realmente faz com que, para você fazer aquele produto, você tem que reengenheirar, de fato. É como se estivesse fazendo ele de novo. Portanto, a melhor estratégia para enfrentar os produtos patenteados é o desenvolvimento antecipado de todas as etapas e institutos públicos de pesquisa. E, para isto, é necessário o estudo da síntese do fármaco, seus intermediários, seus fornecedores, os preços do mercado e desenvolver as monografias analíticas. A solução para os países está em que o seu governo tem uma política clara de desenvolver em escala piloto industrial, que é em torno de 20 quilos de produto, todas as etapas para você chegar a uma formulação, desde a os intermediários, a síntese do fármaco e a manipulação desse fármaco, transformando ele num produto acabado. E com isso, você tem uma noção que preço deveria ser, de fato, aquele produto. Isso nós fizemos no ano 2000, quando Efavirenz estava sob patente e Neofinavir estava sob patente. O que Farmanguinhos fez? Farmanguinhos conseguiu comprar no mercado internacional uma quantidade de Efavirenz, conseguiu já matéria-prima final, não matéria-prima, não intermediário e sintetizar. Infelizmente, Farmanguinhos não fazia síntese de produto, eu só fazia formulação. Então, nós conseguimos comprar o Efavirenz matéria-prima, nós formulamos, transformamos aquela matéria-prima em produto acabado, desenvolvemos monografia analítica, fizemos análise de custo e mostramos ao ministro da época, de um governo que não era um governo progressista como nós temos hoje, mostramos ao ministro o seguinte: ministro, esse produto pode custar 50% mais barato. Ele chamou a Merck e falou: dona Merck, esse produto pode custar, de acordo com a direção de Farmanguinho, 50% mais barato. Isso foi na minha frente. E a Merck falou: não vou abaixar o preço. Ele falou: então a gente vai ser obrigado a entrar com licença compulsória por abuso de preço. E aí a Merck foi embora, tá entendendo? E voltou depois e abaixou o preço. E com o Neofinavir foi feita a mesma coisa. Então, isso é uma estratégia de governo. Isso é uma estratégia fundamental que governos têm que adotar para poder a gente realmente pensar numa coisa importante: saúde não é comércio. É necessário lembrar que o bem-estar da humanidade está acima de qualquer interesse econômico e que não existe maior valor do que a vida. E para isso que a gente está lutando até hoje, para que se valorize a vida e se defenda que patente é incompatível com direitos humanos preconizado pela Grande Convenção de Direitos Humanos. Todo ser humano tem direito a ter o direito a viver. Para isso, precisa ter acesso à prevenção, a diagnóstico e a tratamento. Eu acredito que esse é o último slide antes de passar para o Fortunak. Era o último, não?
Sim, depois inicia a apropriação de custos.
Ok,
Obrigada, Eloan.
de nada.
Professor Fortunak, passo a palavra para o senhor.
Hello. May I switch over to sharing my screen now?
Yes.
Right, bear with me here. This should take just a second. Okay. Do things look fine now?
Yes,
Susanna?
perfect.
Thank you. And first of all, please forgive me that I can't communicate with you in Portuguese. And I'm very grateful for the invitation from the organizers and to my dear old friend Eloan for inviting me to be with you today. I'm here because all of us want to be useful in some way with our lives, and this portion of my life I'm spending with helping, I hope, to assist in making medicines more available to people. I decided to begin with a picture of myself. I hope you can see my cursor over on the right-hand side here. Yes, this is me. I'm wearing a suit and a tie, and this is indeed a nun. This picture is from Tanzania. where I teach a program of industrial pharmacy and drug production at the Kilimanjaro School of Pharmacy. You can't see the picture well in the background, but Mount Kilimanjaro is just back here behind us. To begin today's discussion, first of all, I'm from Howard University in Washington, D.C. Howard University is what is known as an HBCU. It's a historically Black university that was founded shortly after the U.S. Civil War in order to teach freed slaves. So it is entirely fitting, and it's consistent with the mission of the university to assist with addressing unjust inequalities in the world. And this certainly goes along with what Professor Eloan has been speaking about so far today. And I must thank Eloan as we begin this. I met Eloan for the first time, and she probably does not even remember when we first met, the first day that I was working at GlaxoSmithKline in July of 1983. It shows how old I am. But that was a little while ago. And I had the great fortune to be hired by a chemist who was also a woman. And she said to me, there is someone who is quite special who's giving a lecture today who's coming in from Brazil. And that was Eloan. She said, listen to this, what this lady says, because you may begin by thinking that she's crazy, but then by the time she's finished speaking, you may think she's not so crazy after all. And we had a very short discussion afterwards, and that helped shape my life. And so I still always have to thank her for what she's done for me. Now, I believe that you have heard most of this. The one thing that is a little bit relevant, perhaps, to today's discussion is that during my career in big pharmaceutical companies, I helped launch 15 new drugs and somewhere more than 25 generic products, but it's the sales number that is almost staggering here. $250 billion US. Now, a lot of that money, in fact, almost $200 billion of it, resides in one drug, Humira, that is a biologic, that is an immunosuppressive drug that is used for at least a dozen different indications. But my expertise and the way that I can be of service is particularly in assisting how to do manufacturing, and again, in particular, local manufacturing of medicines, and specifically with an emphasis on active pharmaceutical ingredients. I also have a lot of experience in patent litigation because I've learned over the years that patents are a serious impediment so that people die because of lack of access to medicines, and that patents are a reason for this. It was, oh goodness, forgive me, but I'll tell a little story. La Rochefoucauld was an advisor to Louis XVI and to the old regime in France at the time of the French Revolution. And he was quoted as saying, men are not hung for stealing horses, they are hung so that horses may not be stolen. Well, patents were instituted to help societies and to help people. And monopolies were granted to provide incentives, adequate and proper incentives for bringing new and important inventions to the public for the public good. But where the monopolies that are granted by patents result in people dying, this should not happen. This should not be an impediment to access to medicines. And this is why the World Intellectual Property Organization and the Treaty Respecting Intellectual Property Rights are in being. But we'll discuss more of this. But one thing that I ask you for is that, because of course I don't know any Portuguese, I do need help from you. I'll be very, very happy to entertain and to answer as best I can any questions, not just during the presentation, but also as a follow-up. Even after tomorrow, if we can share these by email or some other means of communication, and we can establish a way of speaking to each other to assist you in what your responsibilities are as you are patriots to your country and to helping increase access to medicines so that people don't die. So that really summarizes what the challenges are for me in speaking with you today. Again, I'm so grateful for the organizers for this invitation and for Eloan for arranging this, but I have a limited time with you today and tomorrow. And I am assuming that you're familiar with drug molecules and their structures to some degree. But I also am aware that few, if any of you, will probably have ever made an active pharmaceutical ingredient. But some of you almost certainly have responsibilities for working in drug registration or the regulation or manufacturing of finished dosage forms, the procurement of APIs, and inspecting production and assuring the quality of medicines. I have quite a number of overheads to present to you this afternoon, but many of these I don't necessarily intend that we will speak to in any detail. And I've put them there for your consideration with the hopes that if they raise questions in your mind, that they'll illustrate a little better some of the points that I'm trying to make today that are a summary at a high level, and they may invite you to come back with a question that's more important after you've had some chances to think about what this afternoon's presentation is. So I won't actually read to you the contents topic by topic of what we'll work with together this afternoon, but I will invite you at this point to work with the organizers so that if you can submit questions either orally or by chat, by raising your hand, they can translate those questions back to English for me, and I can make an attempt to answer them for you, or I can follow up with more information. I'll begin by very broadly outlining what it is that contributes to the cost of manufacturing active pharmaceutical ingredients. And I'll split it into two very large buckets. One of them on the bottom here is the investment. It's a one-time investment, or it may be multiple investments, but it is a non-recurring investment in developing a process, learning how to scale up, and demonstrating the bioequivalence, the equivalence and the similarity of APIs and of finished dosage forms, and to a generic company that lives by launching generic drugs. This is the investment that happens with their scientists in achieving a manufacturing process and in filing it with the regulatory authorities. Now, a large Indian generics firm, say Aurobindo or Hetero, Mylan, Viatris is their name now, or Cipla, for instance, estimates that this may be in US dollars, and I'll speak in US dollars solely for this afternoon's discussion, that this may be somewhere between $600,000 and $1.2 million, including the cost of registering in many individual countries. But another part of contributing to the price and to the cost manufacturing of APIs is recurring expenses. And I'll break those into a few different contributions. One of them is the cost of raw materials. Another is the cost of overhead and labor. And a third one is of assuring the quality of what's being manufactured. So that includes good manufacturing practice, quality assurance, the ongoing quality control for each batch production, And this overlaps to some extent with the non-recurring investment of filing and abbreviated marketing application or of a drug master file. But this is the part that is recurring, and it becomes a component of something called overhead and labor. So the overhead and labor here, I have lumped together into the sum of what is it that people make for working in the company who are doing the manufacturing? What is the capital investment in building the facility and in putting in the equipment and the large and small equipment that has a lifetime that is longer than 1 year? So it's not a disposable charge, but it may be 5, 10, 15, 20 years. And a second part of this overhead and labor is the cost of running the facility. So this is generally expressed by companies as an hourly charge. It says, if it costs me $2 million a year to run the facility, I'll look at how many hours that I need to charge that over, and I'll break it down into an hourly rate. So the regulatory component that accompanies the manufacturing is a component of this OHL. The cost of raw materials includes the starting materials, the prices and the amounts, and the yields. So it's the cost of the raw materials and the efficiency of going from the starting material to the desired product. Here's a quick, short illustration of how this comes together to be looking at a cost projection of what APIs cost to manufacture. This was taken from something that it seems probably will not be published. It's a project that I was working on with the TB Alliance for a relatively new tuberculosis drug called Protominid. And the current cost of goods was estimated based upon the scale and the efficiencies and the batch size, along with the overhead and labor for the existing scale of manufacturing. And this was on batch sizes of approximately 100 kilos. You can see the impact of scale. Scale greatly reduces as demand increases, batch sizes get larger, and the equipment in which batches are made gets larger. So the hourly component of the overhead goes down greatly, and the impact of scale can be seen here, decreasing the cost of the API from approximately $3,600 a kilo to about $2,700. In this third bar here of the graph, we have an estimate, a projection of the raw materials cost of production. As the scale demand for an API increases, the pricing per kilo of raw materials goes down. And as the process becomes more efficient, that contribution decreases and has another impact on the API pricing. And this final bar is a projection of using some modest improvements in the yields and the efficiency of a synthesis. And you can see as all of these come together, it is routine for a route of synthesis to decrease the cost of API. In this case, you can see a projection decreasing from $3,700 a kilo to about $1,500. And this is, I'll make a point, it's not for a very large demand drug. It's for drug resistance TB, multi-drug resistant. So this is not an HIV drug. This is a drug for which the ultimate demand might be 10 metric tons per year. But as we'll see, many of the HIV drugs that we're speaking about this afternoon the volume demand is 100 times this, more than 1,000 metric tons per year. Before we begin to discuss the individual APIs, I do need to emphasize the introduction that we heard from Eloan. patents. Patents do have an impact on active pharmaceutical ingredients, and they're a really important means of maintaining market monopolies, keeping prices high, and keeping the revenue maximized for big pharma companies. But also, this impact is that people, particularly in middle-income countries, die. So with low-income countries, and we're speaking about HIV or malaria or tuberculosis, that's a different story. There's the Global Fund. There are mechanisms for voluntary licensing. Low-income countries have a large advantage over middle-income countries in this regard. And I'm not qualified to speak extemporaneously on a lot of the impact of patents and access to medicines, but I do work with an organization called the Initiative for Medicines Access and Knowledge, and you can reference much of what we've published at this website here. But I can speak to the patent strategies for small molecule APIs, and indeed to a certain extent for biologics at big pharmaceutical companies. So originator companies that do drug discovery and that are the first to launch. So a drug molecule is first patented at about the time that the structure of the molecule is known, preclinical toxicology has been carried out, there's been a rudimentary formulation, and an investigational new drug application has been filed with a regulatory agency. So that enables the first entry into humans that I am giving the acronym of FIH. But follow-on patents are a strategy that is assiduously followed by big pharma companies because they want the money. Companies are not charities. They have investors. They feel that their primary obligation is to maximize value to investors. I've been through this discussion many times with the people that I worked with in big pharma. Follow-on patents were very important to us for maximizing the term of our monopoly. And with small molecule APIs, these follow a certain pattern. The first patent covers just the drug molecule itself, but it also covers similar molecules. We needed to exclude other people from working in that same space. So we didn't patent one drug molecule, we patent what's called a Marcouche group. And the Marcouche group would break the entire molecule up into certain modular pieces. Each modular piece would be described in terms of the scope with which we believed we would own the concept of building a molecule using the variety of how large that module might be. So we might have 4 or 5 different modules that the drug was composed of. And we would describe the scope of what we wanted to cover for each module. So the Marcouche group of a small molecule might be very large indeed. It could cover, theoretically, not just a few thousand, but billions of molecules. There are probably about 150 million organic molecules that are known throughout the history of the chemical sciences. And so a Marcouche group can easily describe a potential list of compounds that is far larger than the sum total of all organic molecules that have ever been made. In addition to this, we would follow on with a method of use. Well, you might find that that's a little bit ridiculous in the sense that you invented the drug with a particular method of use in mind, such as HIV. But the way the patent world works, somehow, even to the smart people who are inventors on patents, once we find that it actually is good for the treatment of human HIV infection, you get another patent on that. We would follow that with another patent that would describe the actual dose. So there's a certain culture to this, and these are called selection patents. So the first patent that describes the molecule and its family also would mention, well, we believe that we can formulate this in any way you want. We can make capsules, we can make tablets, we can make solutions, inhalers, all sorts of things. We can dose it anywhere from 0.1 milligram to 1 gram per day. We can use it for HIV. We might be able to use it for other virologic infections. But strangely enough, when we actually found that we had evidence that it could be useful for such a thing, we would file what's called a selection patent. So a selection patent is what we call a method of use, but most particularly, even more so, when we found that within that very broad range for the dose that we had described in the original patent, if we found that the human dose was going to be, for instance, between 50 and 300 milligrams per day, and if it was once a day or twice a day for dosing, we would file another patent. We would say, we're surprised, but within this original range that we put within our claims, 100 milligrams a day and once a day is really good for humans. So we would get another patent. Then we would file patents on the finished dosage form. Now, it's not surprising that oral drugs can be formulated as tablets or as capsules, but every time we made a tablet or a capsule of a new drug, there was some problem to be solved. And patents are based on the idea of there is some inventive step. Now, in the history of formulation science, tablets are made using excipients that are generally regarded as acceptable for use. They're acceptable for use by regulatory agencies. And it's not that huge of a range, but for some reason, every time we formulated a tablet or a capsule, there was a problem to be overcome, and therefore it amounted to an inventive step, and we could file formulation patent. And the API process. Well, chemistry is chemistry. I would make molecules using, very rarely did I invent a new chemical reaction to make a small molecule API. But there was always some combination of steps that made it inventive, and we would say unpredictable, and therefore we would file patents on the process in order to exclude people from using that same process of manufacturing, with the idea that we could keep our competition out of the market because it would be more expensive for them to make their API. I learned over time that people are very clever, and there are a lot of chemists in the world, and API processes are actually a wonderful way of stimulating innovation that brings down the prices of APIs. But one thing, a friend of mine at Purdue University, once upon a time, maybe about 35 years ago, convinced a federal court judge in the United States that crystalline forms, organic molecules, are sort of floppy. And when it comes to the molecules, well, molecules can exist in the form of a gas or a liquid or a solid. In the gas phase, the molecules are bouncing around in the vapor. If we reduce their molecular motion, they condense down together and they become liquids. If we reduce the molecular motion even further, and there is very little freedom of movement of those molecules, they become solids. And in the solid form, molecules tend to pack together closely, and they pack up against each other they like to make a regular packing arrangement in which the molecules will have a regular three-dimensional array of their crystalline structure. That three-dimensional array makes them crystalline. Because that packing is largely unpredictable, it was judged to be inventive. And those packing arrangements, the different crystalline forms, salt forms, co-crystals, solvates, they can be patented. And this is a really difficult. I admire the countries who've said that crystalline forms, because they are inherent property of the molecule itself, are not inventive. But that's a different story and a different discussion. But another way of extending monopolies is additional methods of use. And that is very often companies you need to get to the market as quickly as you can. The company has invested money. You want to make a profit based on your investment. So you pursue the clinical approvals that are the fastest that you can get approval for. But then you follow on. I don't want to waste a molecule. It has a certain mechanism of action. If that mechanism of action and interplay with biological proteins can allow me to have more than one method of use, then I must pursue those additional methods of use. And when you finally demonstrate some clinical utility in additional disease states, then you file for another method of use. For small molecule APIs for HIV, it happens, but it's relatively uncommon. Tenofovir, for instance, is good not just for HIV, but for hepatitis B. It's relatively uncommon for HIV drugs to be good for multiple different diseases, but it is very common, for instance, for cancer drugs to be applicable to more than one different type of cancer. But additional methods of use also cover combinations of multiple drugs. And this is where we're getting to the boundary between what might very well be described as obvious to combine multiple drugs to treat HIV versus the idea of, well, I invented dolutegravir, and I would never expect it to be good in combination with tenofovir because my drug is from GSK and tenofovir is from Gilead Sciences. Well, patents have a certain rhythm to them. They are written after a fashion a little bit like poetry. It's something that approaches perhaps iambic pentameter. So patents are meant to have a certain structure. There's an abstract, there's a summary, there's a specification that discusses what's known in the prior art, the background and how the idea came into being and what the inventive step is, what the problem is to be solved. But patents also must have examples of the invention itself that disclose in sufficient detail how a person of normal skill in the art, this is often summarized at the acronym as a POSA, of how to reproduce the invention. But this is an antagonism with something that is the manufacturing process. For many different reasons, a process patent never reveals an actual manufacturing process. It may be possible in some process patents that they might choose to reveal the actual manufacturing process, but it doesn't happen. Look, patents are almost always filed. You need to protect your invention before you actually use it for manufacturing. So patents are filed before commercial manufacture and the validation of a process. So that final information isn't in the hands of the inventors when the patent is filed. In addition to this, the scope of any single invention largely does not cover an entire process of manufacturing. And the actual analytical methods, final specifications for an API and a finished dosage form. So the impurities. The development report describes how the manufacturing process was arrived at, how you assess the risk in each stage of manufacturing, and how it's managed. How you validated the process to assure the correct and allowable ranges for each critical parameter of manufacturing. And those details, they just are not contained in patents. It makes no sense for a company to reveal those. It would just be giving away to your competitors details that you don't want them to know because it's a competitive circumstance. So with that, let me begin to discuss a little more information about specific active pharmaceutical ingredients. And I'll begin by just discussing a little bit about active pharmaceutical ingredients in general. Okay, APIs, those are the active molecules in a finished dose form that cause the intended therapeutic effect. So dolutegravir, for instance, may be delivered as a 50 milligram tablet, or it may be delivered as a depot formulation in combination with rilpivirine, or it might be delivered in a triple combination or a double combination in another fixed dosage form. In the history of medicinal chemistry and drug discovery, there are a little over 16,000 drug molecules that are listed in the drug bank. Now, out of these, though, there are less than 5,000 APIs that have been approved for human or veterinary use in animals worldwide throughout history. The World Health Organization has the essential medicines list, and updated as of 2023, this contains 591 drugs and 103 additional therapeutic equivalents. Drugs, so these active pharmaceutical ingredients, they can come from natural sources, from plants or animals. They can come by synthesis, so they're made from simpler starting materials. They can be made by biotechnology or by any combination of these. And you're fully aware of these. For instance, nowadays, there's a lot of emphasis on drug antibody conjugates, so that a small molecule conjugated to an antibody to increase the efficacy of both approaches to treating a therapeutic disease, for instance, cancer. Active pharmaceutical ingredients are regulated. They have roughly the same regulatory requirements as finished dosage forms. And to me, of course, because they're more basic, I feel that it's just as important regulating those, even though some parts of the industry, the people who deal with primarily with finished dosage forms, can look at APIs and say, that's a raw material for my process. I don't really need to control it as quite a high level as a finished dosage form. Well, for those of you who aren't yet familiar with this, the guidelines that manage the regulation of APIs and finished dosage forms, they're available from the International Conference on the Harmonization of Requirements for Drugs for Veterinary and Human Use. This is called the ICH. The ICH is a wonderful organization. It was founded in 1995. It was believed that it would only be in existence for 10 years. But one thing that is very useful is to look at the ICH quality modules, ICHQ. These are 14 different documents. There are 14 different modules that deal with different aspects of achieving and controlling quality in drug manufacturing and control and delivery. They're available at this website. They deal with, for instance, things like stability. And ICH Q7 and Q11 specifically deal only with active pharmaceutical ingredients. Now, I've dealt with Anvisa with regulatory filings, but not nearly as much as I've dealt with, for instance, the US FDA or regulatory agencies even in India, China, and Africa, and the European Union. But Anvisa has some differences with international requirements, but it is a strict regulatory authority for the purposes of regulatory filings and for good manufacturing practices. So to transition us a little bit now, I'm showing you a picture. This is actually posted online. I downloaded it today. It's from some friends. I actually know this person whose picture is right here. They're people at a really good generic company in India called Loris Labs. And I'll speak a little bit more about them as we continue onwards today, but it's of interest. This is what an active pharmaceutical ingredients manufacturing plant looks like. This looks very busy, doesn't it? This looks very different from a finished dosage form manufacturing suite that's making tablets or capsules or oral suspensions. And it's because there's an awful lot of different utilities and peripherals that are attached to these manufacturing vessels. So making small molecule active pharmaceutical ingredients by chemical synthesis is what we're seeing. These reactors, these are about 4,000 liters each. And you need a solvent as a heat sink and as something to help moderate and promote a small molecule, a step in a small molecule synthesis of breaking old bonds and making new ones to start from simple molecules with multiple purposes that are inexpensive to go to much more complex molecules that are the API. That's why these vessels are of such large size. And in a 4,000 liter reactor, you can only get it about 85% full because it needs to be stirred and it needs to be heated and cooled and solvent needs to be distilled. And with that 85% fill rate, you can put maybe 300 kilos of product that will come out of each step of a synthesis in a reactor this size. What you're seeing here is this is an agitator. These large pieces of equipment at the top of the reactor. This portal here has got a glass that the operator can look through to see what's going on. And these things, this piping here, is things like an inert atmosphere, nitrogen gas, vacuum, distillation takeoffs, and pipes for additions and for removals. And you can see valves everywhere to open and close things. And what you can't really see, but you should be able to see, is that these should always be clearly labeled so that you can understand, the operator can know what it is that each one of these is for so that they won't make a mistake during operations. Well, the general quality requirements for making APIs are that the facility itself needs to be designed and built and maintained to meet good manufacturing practice requirements. The actual manufacturing, the systems under which the facility operates, has to be managed by a QMS, a quality management system, that is responsible for all of the relevant systems of the procurement and the receipt and the testing and approval of raw materials, warehousing, the actual manufacturing, testing in process and finished product, and storage and stability and packaging. All of these systems are managed by the QMS, and the management system has the final say on whether or not a product is allowed to be sold. The owner of the facility cannot override the responsibility of the quality unit to make final decisions about the approvability of all products. Now, we get into this business of China and India now. China and India make most of the world's active pharmaceutical ingredients. And it should be kind of, it's obvious, but you may never have thought of this before, but an API, where it's manufactured, it must be acceptable for use. And in the country who's purchasing it, it must also be acceptable for use. So APIs are not just made under good manufacturing practice, but the process is validated. It's filed with the regulatory authorities, and the facility and the filing are approved by the National Drug Regulatory Agency in the country of origin and in the country of use. Companies that make APIs for multiple purposes, they have to be able to meet the requirements of all of the countries that they're selling to, but they also need to sell their APIs to multiple different end users. So a drug master file is the typical regulatory filing for APIs. And a drug master file, it is something that is kept secret. It's not part of the marketing approval. A company that makes an API files a drug master file, say in Brazil. And the drug master file is not reviewed by the regulatory agency until a finished dosage form company in Brazil references that drug master file. After they reference it, the regulatory agency looks at the drug master file and combines it with the marketing application and says, yes, that API is made in a particular way that makes it acceptable for use in that finished dosage form. When it comes to how you make APIs, quality management system, it probably makes up 20%. When I was in the United States, the QMS was about 30% of the total headcount of the manufacturing organization. When you speak about an overhead and labor rate, an hourly charge for running this facility to make APIs, it's generally about $700 an hour for a generic company in India. And if you're making on this scale a single step of an API synthesis, it is almost impossible to do more than one step per day. Even if the reaction itself only takes 1 hour, staging all of the raw materials, charging them into the reactor, achieving the appropriate conditions, working it up and isolating the product, it takes a full day. Companies typically work 6 days a week and 2 working shifts, so 16 hours a day. Now, if we consider that earlier picture of a manufacturing train at Loris, it had multiple different reactors in the same train. So a company might be making 3 batches of the same step on the same day for that same amount, a single overhead and labor rate, or they might be doing more than 1 step of the process in the same train if that's validated. So that is how companies tend to try and maximize their efficiency in terms of overhead and labor rates and that contribution to API manufacturing cost. So you'll forgive me if I've been a little bit perfunctory, and if I've been very high level about speaking of those particulars for API manufacturing. But now let's start looking at the individual APIs for use themselves. And you can see them here. We're looking at these major APIs. So we have dolutegravir, which, in my world, when I'm working with the Global Fund or with USAID, who are funding antiretroviral treatments in low-income countries, dolutegravir is the backbone of once daily oral ART. Tenofovir disoproxil fumarate is another huge drug that it's along with dolutegravir is the most heavily used by the most number of patients. Tenofovir alafenamide fumarate is a close analog, but both of these are prodrugs of the drug itself, tenofovir. Tenofovir is the intracellular active anti-HIV agent, but there is a substantial difference between these two. Tenofovir disoproxil is 300 milligrams a day, while tenofovir alafenamide is only 25 milligrams a day. But as you'll see, it's debatable. This tenofovir alafenamide, or TAF, has a real challenge and has not been able to replace TDF, even at the much lower dose. So a combination of dolutegravir, which is an HIV integrase inhibitor with one mechanism of action, and one of the tenofovirs, which is a nucleoside reverse transcriptase inhibitor, so it affects another protein in the life cycle of HIV, and a third drug with a third mechanism of action, a non-nucleotide reverse transcriptase inhibitor, either lamivudine or emtricitabine. So one of each of these, or three drugs, is the typical oral ART. We're also going to speak about atazanavir, which is the primary protease inhibitor for second-line treatment. Efavirenz, that has very substantially been changed out for dolutegravir since about 2017, although there's still a significant amount of demand. But what we won't speak about today is two drugs where there isn't a whole lot of ability to predict the pricing and the usage of these yet, because they're components of these long-lasting depot formulations. So cabotegravir is an integrase inhibitor just like dolutegravir, but with a slightly different structure, and rilpivirine. But I'm a chemist, so you forgive me. I want to show you structures. Here are the different structures that we have, but showing you the structures actually reinforces what I mentioned on the previous slide. So here is the structure of dolutegravir as an integrase inhibitor. If I showed you this and overlaid it with other integrase inhibitors, like cabotegravir, bictegravir, raltegravir, and elvitegravir, you would see some significant structural similarity. But where the similarity really strikes home is if we look at tenofovir disoproxil fumarate versus tenofovir alafenamide fumarate. Now, I have fumarate in parentheses here because I'm not showing the fumaric acid that comprises the salt. This is just the structure of the individual molecules. This phosphorus right here is a key. So this is actually a very simple structure for a drug molecule. With the phosphorus, it mimics the action of a component that is absolutely needed for incorporation into a growing HIV viral particle. And when this active drug is incorporated into a replicating virus, it stops the replication cycle. This spinach in here that hangs off of the phosphorus, you'll notice how this is on the left-hand side for tenofovir disoproxil versus tenofovir alafenamide. Those are totally different. But each one of these drugs is an analog of the basic molecule itself that I'm circling with my cursor both here and on the bottom. You'll see that those components of the drug are in common. So they are analogs of each other. Now, forgive me, I need to go back. Interestingly enough, when tenofovir was first invented, it was appreciated that it was a stunningly potent antiviral compound, but it was a total failure from a kind of a medicinal chemistry approach. It was so water soluble that it couldn't actually get into an HIV-infected cell. If it was dosed into an animal, a rat, a human being, the actual amount of drug that got into the infected cell was very tiny. And these prodrug pieces needed to be put onto the antiviral part in order to make these drugs more lipophilic, more oil-loving, or more loving fats. That way, they had the right physical properties so that they could be transported across cell membranes so that when a human being took them orally, it could actually be bioavailable, which is the right term, so that it could be delivered to the HIV-infected cells. Between these two, the prodrug portion of TAF is not only substantially different from tenofovir disoproxil, but it was cleverly designed so that an enzyme inside the cell removes these prodrug pieces. That is why the dose of TAF can be so much lower than the dose of TDF. However, we'll also see these two drugs, TAF doesn't seem to have any clinical advantages over TDF. Okay, but now let's look at lamivudine and emtricitabine. My goodness, look at these. Where's the difference? If you're not clever, you could be looking at these structures and you'll miss the fact that the difference between lamivudine and emtricitabine is right here. Lamivudine does not have the fluorine right here that emtricitabine does. So the acronyms for these molecules are actually 3TC and FTC. They're so structurally similar. Emtricitabine is moderately superior to lamivudine in HIV assays in a test tube. But interestingly enough, there has never been a clinical trial published that has clearly demonstrated that a combination of drugs for HIV, if the difference between two combinations is simply changing out lamivudine for emtricitabine, there's no difference in clinical efficacy, no difference in the ability to suppress viral replication. Nevirapine was an early non-nucleoside. So lamivudine and emtricitabine are nucleosides by virtue of this structure. They have a nucleoside here and they have a sugar ring here. So these are inhibitors of HIV reverse transcriptase. Nevirapine is also an inhibitor of HIV reverse transcriptase, but it's a uniquely viral enzyme. Human beings do not have a reverse transcriptase enzyme. So because that enzyme is unique to HIV, these drugs are particularly good for HIV. But nevirapine acts at a different place, a different portion of that reverse transcriptase enzyme. So nevirapine came along, and in combination with nucleoside drugs, it was very effective. And a combination of 3 drugs containing nevirapine was really good for inhibiting, once daily antiretroviral treatment. But nevirapine was largely changed out first for efavirenz, and then secondly for dolutegravir. So the current demand for nevirapine, it exists, but it's a sort of a specialized need. We'll also speak about efavirenz, as I said, and atazanavir as a protease inhibitor. So that's a different mechanism of action once again. Okay. Now I'll pull back for a second from those HIV molecules that we've been discussing, and I'll get us into how the routes of synthesis of small molecule APIs. It's not always the case, but usually the starting materials have multiple uses, and so they're purchased, and occasionally there's starting materials that only have use for making a drug, but they have to be available. This is what the regulatory agencies insist upon. These have to be available for purchase. They have to be well characterized, and they do often have uses other than in API synthesis. You start from those starting materials, right? You, as the manufacturer, have a duty to the regulatory agency and to the public. You must justify your selection of what the starting materials are. And this is based upon your responsible knowledge. How do I know that if I begin with these starting materials that are purchased with certain specifications, as I go forward with good manufacturing practice, if I do this, if I purchase according to the specifications and I manufacture under good manufacturing practice, the API will meet its intended requirements for quality to assure that it's pure, it's safe, and it's effective, and it's bioequivalent. I take those starting materials and I break old bonds and I make new ones. So I'm usually increasing the molecular complexity in successive steps. Now, one thing that is often difficult to appreciate is that drug molecules, they have multiple routes of synthesis. When you're first discovering a drug and you're getting it through human clinical trials, you don't particularly care if it is the most efficient synthesis. You needed to get it into assays to test it. You need to test its preclinical pharmacology and pharmacokinetics and its toxicology. Then you need to get into human clinical trials. Speed matters more than money under these circumstances. But when the drug is coming to the point where it needs to be approved for marketing and for human use, that's where you need a commercial route of synthesis. So that's what needs to be under current good manufacturing practice. It needs to be validated. So we know that validation is a process that says, I understand the critical parameters, I have them outlined, and if someone of normal skill operates the manufacturing process as it's written, I should reliably and reproducibly obtain API that meets all of its quality requirements. That's process validation. But the synthesis must be practiced under good manufacturing practice, and I need to have a quality assurance unit. And I also need to affirm that the API that I am making, no matter what the synthesis is, it is equivalent in terms of its bioavailability. That usually means particle size, distribution, and crystalline form to assure that it dissolves and is absorbed and distributed throughout the body within what's called the window of distribution. Well, this is relevant for generic production of APIs because those originator companies have taken the opportunity to patent the processes of making APIs and of finished dosage forms. So the route of synthesis is very often different for a generic company versus the originator. Well, because that generic company most often has to avoid infringing, they have to look at those process patents and say, oh, goodness, I see that inventive step. I'm not going to infringe on the claims for the scope of that invention, but I'm going to cleverly come up with something else. But where this gets even more complex is that generic providers very often patent their route of synthesis. So if there are 15 generic companies making lamivudine, there are 15 different process patents for each of those APIs. Or at least there may be. Okay, now let me show you, to start us off, some molecules. So you will forgive me being a chemist, please. I'm not trying to make you an expert in chemistry with this afternoon's presentation. But instead, this is just to illustrate something that will help us through the rest of our discussions. I know this chemistry very well because I launched this chemistry way back in 1997, before some of you were even born. This is the first generation route of synthesis for that HIV drug efavirenz. And so I worked on this at DuPont Pharma. Now, for instance, I personally made 70 tons of this. Look, the point of this is to show you how this is really busy, isn't it? There are a lot of structures here. So I'm starting from one molecule here, and I'm doing some chemistry that's described with these arrows, and I'm showing you how that chemical step transforms one compound into another molecular structure. And you can see how that complexity changes with each step until we arrive at the drug molecule all the way down here on the bottom. I apologize if this is too simple for those of you who really are experts in chemistry. Right. Highlighted in yellow here, 4A and 5A. So that's this molecule here. And I should cross out that lithium. 4A is cyclopropylacetylene. And 5A, without the lithium, is pyrrolidonyl norepinephrine. These are two key starting materials, KSMs, for the first route of synthesis of efavirenz. Now, if you notice this right here, this is a piece that was put on as what's called a protecting group. But you'll see that this piece is put on and it's retained for a couple of steps, but then it's removed. It's not actually part of the drug molecule. That worked really well when we were very excited. We knew we had a good HIV compound that was useful and the world wanted it. But it's not efficient. It's introducing molecules and atoms that we need to get rid of later because we don't need them in the drug. And so because this isn't efficient, it also greatly increases the cost of manufacturing. Eventually, the third route of manufacturing, and I'm going to disclose to you something that's not widely known, is we actually did this route of manufacturing at DuPont pharmaceuticals, but we were being purchased by another company at the time, and they didn't want to go through the regulatory problems of filing this third route of synthesis and getting it approved. So we never even patented that route of synthesis. But we did communicate this to a really good Indian generic company that I mentioned, Loris Labs. And I have to give them a lot of credit because they took this from an idea, from a 1 kilogram idea that had been executed in the lab, and they understood it and modified it so that they owned this, and they came up with their own patent filing so that they do have their own individual patents on this third generation route of synthesis. This is what was used, and we will see the cost of the API from the different generations of synthesis as we go on. We're going from a 6-step synthesis of the API to a 3-step synthesis. And the difference in the cost was really profound. But this, you can see that this is a much less busy overhead than the first route of synthesis. But we also have that third key starting material, compound 9, that's highlighted right here. All right, so you'll forgive me. I hope that I'm not causing you to go to sleep if you're not really interested in chemistry, but please bear with me, if you will. All right, let me go on. Let's start talking about some of these. The first one, I'm going to go into some great details about dolutegravir because it's viewed largely as a real backbone of HIV ART. And look, one thing that I want to describe to you is it's this culture, this poetry of how patent applications work. A person of normal skill in the art reading the process patent that covers the route of synthesis for ViiV. So this is a kind of a murky, multiple companies are involved here. GlaxoSmithKline licensed this structure from Shionogi in Japan. And Shionogi is also part of Japan Tobacco. Well, GlaxoSmithKline largely brought this drug through development, but GSK then partnered with another company, Pfizer, to come up with ViiV Healthcare. And ViiV was partnering their assets in HIV of multiple companies in order to to focus on HIV drug discovery and development. So it's a GSK patent, but dolutegravir is actually owned by ViiV. Well, let's talk a little bit about that process patents to GSK. And I have to give a little bit of credit here. There's someone in here named, as one of the inventors, named Matthew Kowalski. I actually worked for his father when I worked for 10 years at GSK. It's a remarkable sort of thing in the sense that you think about becoming a PhD chemist. Who wants to do that? I'm going to admit to you, when I first went to university, I never wanted to become a chemist. But it must be something genetic because my older brother's a chemist. I went to the university. I knew I was going to fail university chemistry. And so here I am. And Konrad Kowalski used to be my boss, and Matthew Kowalski was his son. And I remember him as a child saying, I'm never going to be a chemist. And yet he became one and worked for the same company as his father. And he's the one who came up with this original process for making dolutegravir. Dolutegravir is not even in the name. It's not even mentioned in this patent. If you search for process patents on dolutegravir, you won't find this one. This is a process for preparing carbamyl-pyridone derivatives and intermediates. A process is described, but it's got flexibility in it that it covers both the syntheses of dolutegravir and bictegravir. You notice these structures are very close to each other? And it illustrates some of the complications of patent chemistry. Along the way, ViiV and GSK launched dolutegravir that's here on the left. Gilead Sciences owned the rights to emtricitabine that they in-licensed from a small company, and tenofovir, and they actually in-licensed tenofovir as well. However, looking at these, you see the structural similarities. You would think that one is obvious from the other, but it's not. At least not Gilead didn't think so. Gilead, independently, without consulting with GSK, brought bictegravir through development into the market. Because of this, the two companies sued each other. GSK said it's obvious that bictegravir is an effective HIV drug based on dolutegravir. And Gilead said, no. Look at the differences between the two molecules. It's clear that bictegravir, nobody looking at dolutegravir would consider bictegravir to be an effective drug. Well, they did settle their differences. And in the end, Gilead Sciences paid royalties for several years to, in fact, I think the royalties end in 2024, to ViiV GSK for having brought bictegravir to the market. But bictegravir is actually a larger sales in HIV in the United States and Europe than dolutegravir is. But to get back to the patent itself, before we discuss some of what's in the patent, as an illustration of what you might take away from today, there are 19 claims in the patent itself. But there are dependent claims and there are independent claims. An independent claim says, this is a significantly inventive component of my invention. And the dependent claims, they depend upon an independent claim. So I'll discuss that a little more when we reach that point. But this patent, it's 19 claims out, 5 independent claims, and 1 claim covers a molecule. The 1 claim that covers a molecule, this is a claimed intermediate compound. This is not the drug. It claims the compound that has the O methyl group right there. So methyl is a CH3. And it also covers the same analog in which this part of the molecule is the same structure for bictegravir. Now, right, when you're filing a process patent, you would like to own the compounds that you use as your intermediates, but you can't do that if they are in the prior art. So in addition to filing patents, reviewers at the patent agencies have to review the claims and approve them. And competitors would like to invalidate patents. There are 2 major ways to invalidate a patent. One is by obviousness, and the other one is by what's called anticipation. Obviousness means a person of ordinary skill in the art would read the invention and look at the claims, and it would be immediately obvious to them. They would say, I know enough general organic synthetic chemistry that it would be obvious that these work. So that's fairly easy to understand. But anticipation means that somewhere in the prior art, that actual invention has either been published or it's been the subject of some other patent claims. Now, I can guarantee you that every time a generic company wants to bring an HIV drug to the market, they get sued by an originator. As long as the originator has any patents that are still valid, they sue them. And it may sound horrible to you that this happens, but what it does is it starts an awful thing called the patent dance. And if you think about dolutegravir, look, dolutegravir, the sales in 2021 were over $4.5 billion US. I mean, that's over $10 million a day. And so if you have any ability to file suit against a generic competitor, you do it. And the reason is that it starts this thing called the patent dance. The patent dance is an automatic 30-month stay that prevents the generic competitor from launching in the United States. At $10 million a day, 30 months is an awfully long time and an awful lot of money. So patent litigation is a way of life for the generics. Let's actually look at the process for dolutegravir/bictegravir. And it's not to make you conversant with the chemistry, but instead to illustrate what goes into the specifics that are revealed in patent filings. So this is the whole route of synthesis that was exemplified, right? So in a patent, you've got an abstract that tells you what the invention is. You've got a specification that outlines the background, the need for the inventive step itself, and the solution. Then you have examples that illustrate the practice of the invention. Then you have the claims. And the claims are what's really important because the claims are in legal terms. What's called it comes from the French, the metes and bounds. So that's the boundaries or the scope of what the claims cover. In the examples of this patent that we're speaking about for dolutegravir, this is the synthesis that was disclosed. But you'll see, look at the little difference right down here. See in this right-hand side, so 1, 2, 3, 4 rings in this molecule. Look at that bottom right-hand ring. That's bictegravir. Let's compare it. Bictegravir here on the right and dolutegravir on the left. This is a 5-membered ring, and this is a 6-membered ring. So the actual examples in the patents described the synthesis of bictegravir. So it tells us that GSK Vive didn't want to, they elected not to disclose the actual synthesis of dolutegravir. The claims of the patent are divided into 3 different stages of the synthesis. So what we're seeing right here is stage 1. So it's going from commercially available starting materials over to intermediate number 3. You can see that that's the first line all the way up here. This is actually amenable to continuous synthesis. And what's really useful is that this and similar compounds are starting materials now for good manufacturing practice. And that has helped greatly decrease the cost of generic API. This is actually pulled directly from the patent itself. This is the example that describes stage 1. Now, I'm not going to ask you to read this, and I won't read it to you. What I'm trying to make the point here is that this is not easy to reproduce. Okay, it tells you the quantities. It says you can mix one starting material and another one and stir it at room temperature for an hour and a half. And then it says you add methanol, you add another reagent, you stir it, then you concentrate it. I wonder what you mean by concentrated. I wonder what you mean by room temperature. And then it says you charge some other things portion-wise. Well, a person of skill in the art doesn't know what's meant by portion-wise. It tells you add lithium hydride, LiH, while maintaining below 25 degrees centigrade, but it doesn't tell you how long. Do you add it over 1 hour, 2 hours, 5 hours? I can personally tell you from people doing this in our laboratory, this is added over several hours. If you just read this, you wouldn't be able to readily reproduce what's happened. Then it says you do some other things, and the main point that I'm getting to is that there are enough details missing from this description that one of skill in the art would have to do a lot of experimentation to reproduce whatever the results are, but you don't even know the results. It says, look, the product was collected by filtration and dried in a vacuum oven, obtained as a solid. It doesn't tell you the yield. You don't know the efficiency. You don't know the purity. And you don't know anything else about the characterization of this intermediate in this process. Here's the second stage of the synthesis. So you're doing some particular things. You've converted what was an ester here to an acid. You turn this into an aldehyde, and then you use this along with another piece to make that bottom ring. Okay, here's that description as a process example in the patent application. Once again, there are a lot of things missing. There's a lot of times, there's a lot of temperatures, there's a lot of in-process controls. So I'll put it this way. When you're doing a synthesis and you scale it up, when you go from a small scale in the laboratory to 10 times that scale or 100 times or 1,000 times that scale, many parameters simply must change because of the laws of engineering and the laws of thermodynamics. And there's no such thing as in-process controls here. How do I test for when a reaction is finished so I know that this is what the inventors were doing? What's the purity of what I got? What's the yield? What's my efficiency? It's just not there. Here's the third stage of the synthesis, putting the rest of the pieces together and making the final molecule. Again, those same sorts of details, forgive me, I'm going to go backwards. Those same sorts of details are missing. Without discussing that further for a moment, but hold it in your mind, I'll go forward. Let's look at claim 1 here. Now, claim 1 is usually the most important claim in a patent. It is the most important independent claim, and it usually, but not always, not in this case, it may describe the full scope of the intended invention. Well, look, I noted that this claim is quite specific in a certain sense. And what it is this: it says it's a method comprising contacting this specific molecule. So I have a methyl group here, a methyl group here, and 2 methyl groups here. And I'm contacting it with what's called an oxalate ester of formula 2, where these 2 R groups, they're not specific. They're general. They can be alkyl, aryl, benzyl. And in the presence of something called M plus OR minus, where M plus is an alkali metal cation, and I'm forming a pyridinone of formula 3. So what we know here, right? This is This is nothing like looking at a huge Marcouche group that describes just, the whole of all of the molecules claimed by GSK Vive when they invented dolutegravir. This is much more narrow. And although the description is broad, you can see that process patents generally have to be very specific because they need to avoid prior art and they need to avoid obviousness in how they execute their chemistry. So let's think about that process patent for a moment. Look, there was enough there. And I've looked through something called the file history of how the reviewers reviewed and the comments that they made on the patent application. It took them 3 tries to get this patent issued. The first time it went in, the reviewers rejected it. It went back a second time, and the reviewers looked at it again, and they rejected it. Then finally, Vive and GSK modified the application a third time, and they were awarded this process invention. Well, the examples were good enough to show that the inventors, they owned the claimed invention. They could make dolutegravir or bictegravir. But there are some arguments in here that this should not have been awarded a patent or that the claims should have been restricted. Because as we've seen from that really broad description of the process, it's not clear how to practice the best mode of the synthesis itself. The rates of addition, times of reaction, reaction endpoints, tests, purity, impurities, degradants, these are all missing. There's no description of a crystalline form or a crystalline process for the API. Now, what came out of all of this is that generic companies developed their own syntheses that got around the GSK patents, that don't infringe any of those 19 claims. And these patents have really reduced the cost of production of that API. Now, I'm going to refer to a publication that, in fact, I have gathered up all of the materials that I've referenced, and I can make these available to the organizers or to you. I should have put my email along with this presentation so that you could contact me individually if you like. I'll be happy to provide to you either a copy of the overheads. I'm very happy that this is being recorded and available to everyone. And I can also make available to the organizers or to you all of the materials that I used in preparing this presentation. This is a really good review of the patent literature and also some of the journal manufacturing, the journal chemistry. In the chemical publications, in the chemical art, if you're working in a company and you want to actually make an API, you largely refer to the patent literature. That's because it describes on what people think is important enough to assert ownership. It's important enough that you spend the money and you go through the effort to file claims and to get those claims issued. It means you think it has commercial value. So everybody, or almost everybody, who is practicing this synthesis now has their own process patent. And that reveals their thought processes and their innovation for the synthesis of dolutegravir. Journal articles that are filed by, that are published by academics, people like me, they can be useful. But if you look through the journal publications on dolutegravir, what you find is an awful lot of academics are actually only catching up to what the generic producers have been doing for 5 or 10 years already. It's an odd kind of a thing, but it's interesting. And I've worked a lot with a Chinese generic company called Desano, And once upon a time, we were having we were having a little bit of relaxation before dinner. And they said, well, let's use that typical phrase on you. Sometimes people think that only white men can think. Well, there's another part to this as well. Sometimes people think that only the originator company can think. Generic companies have their own requirements. They're really smart. And they make their living based on having better formulations and better chemistry. So they are really clever and innovative. And that's why the patent literature is a really useful place to go. Now, how all of this is available to us today instead of 10 years from now comes from the medicines patent pool. The medicines patent pool negotiated a license with ViiV. And ViiV then, for very, very low percentage royalties, maybe up to about a 5% royalty, said, look, I have patents and I insist on being able to sell my product to high-income countries and most middle-income countries. But since I won't make any money in low-income countries, I'll license this to a generic company, and you can file and get regulatory approval at your expense. And in addition to this, what I'll let you do is you can use whatever you want. You can use my drug. And if you want, I'll even let you license my route of synthesis. So I'll provide a technology transfer package to you, and you can make the API the same way I made it. Well, almost all of the companies that negotiated a license with the medicines patent pool came up with their own synthesis. And they said, wait a minute, I'm not going to infringe on your patent application. I've got my own process, and my process, I think, is better than your process. So that teaches us something. Those people are looking at the way they manufacture things in their manufacturing plants, and they're saying, I think I can beat the price of what ViiV manufactures the API, and I'm going to use my own chemistry. Well, here, the daily dose of dolutegravir sodium is 50 milligrams. So a patient needs about 18 grams of API per year. Now, generic companies launched dolutegravir for low-income countries with really small production volumes. There's a certain process about how this happens. You want to be small scale and reduce your risk and your expense until you see if there's product demand. But they were making 10 and 20 kilo batches, about 1/10 of commercial size when they first launched. So they did formulation, they did bioequivalence, and they were going according to regulatory guidelines about scale. So they launched their API at about $4,000 a kilo. So that's $4 a gram. And $4 a gram times 18 is $72. So that would be the cost to the company of the API that goes into the formulation. But after that, when demand increased and when people got a chance to scale up their process and understand it and improve it, the API price rapidly declined. And this is what chemistry folks in Brazil would naturally do and have done. It's routine. Okay, but now is where I need to talk about something called data mining. So it's not easy to get data on what APIs cost. We're interested in two things. One is, what's the cost of manufacturing? The other one is, if I could buy it, what price can I buy it for? Well, one thing we can get at, we have an idea of what kind of a profit margin companies need to make on APIs. That's typically between 20 and 30%. But there is one really reliable way, but you have to pay money to do it, to get data on what APIs are sold for as a price. That's import-export data. So as I said much earlier, China sells a lot of APIs to India. And we are really fortunate with dolutegravir because there are a couple of really good HIV companies. China companies, they tend to specialize, right? Those of you who deal with this area, you know that. Indian generic companies tend to be very broad. They'll launch hundreds of products across many therapeutic areas. Chinese companies tend to cluster. One company will focus on making fermentation rifamycin products. Another will do beta-lactam antibiotics. The third will do HIV drugs. Well, we've got Amneal and especially Desano. Desano's a really terrific company historically for making HIV APIs and then selling them to India. The chemists at Desano are really excellent. And in addition to that, they'll talk to you. They'll communicate back and forth. And it's their data that contributed a lot to the information that we're going to discuss here. Well, they can't give us that data because internally that's a secret. We have to pay for it. We have to buy import-export data. All right, where did we get this from? So mostly a lot of this information came from a place called the Trade Vision. You can find them online. You can also find Panjiva. Panjiva is a source from the Indian government. Panjiva used to publish the information publicly and free of charge until the end of 2016. But then Indian manufacturers started to complain that it was revealing too much confidential information, so they stopped providing it publicly. And that old information is available in a place called PharmaCompass. And PharmaCompass has little snippets of newer information in it. Panjiva can still be purchased, but this is $9,000 US per year. That's a lot of money. I don't know about you, but I can't afford that. Anyway, this is the sources for where we got our information. Okay, let's look at what the numbers say for what dolutegravir raw materials cost, okay? There are 4 different raw materials that are expensive, that substantially contribute to the cost of the API, and they're listed here. The product from the stage 1 that we were discussing, let me see, do I have those structures? No, I don't. Okay. This is the compound that was labeled as 3 in the stage 1 of the synthesis. This 2,4-difluorobenzylamine goes into stage 3 of the synthesis. And R3-amino-1-butanol is the key intermediate in stage 2. And ethyl 4-methoxyacetoacetate, it's not hard to make, but there isn't much demand for it, so it is much more expensive than it might be otherwise. All right, now I'm going to refer to this. I'm going to rapidly go back, okay, to what we started with in this synthesis several overheads ago. Stage 1. This is the pyridone over here, 3. These original steps were done by GSK under good manufacturing practice. But nowadays, this material moves in commerce, and it is the starting point for GMP. That decreases the cost, because when you shift away from a GMP manufacturing plant, you don't have to have the same quality management system. And the whole price of operating probably decreases by more than 50%. This piece right here, this is 2,4-difluorobenzylamine. And this is an aminopropanol. It's a similar piece that goes into dolutegravir. All right, let's look at what happened to these starting materials over time. So the pyridone price decreased from $400 a kilo in 2018 to about $80 a kilo today. That chirol, the 3-amino-1-butanol, has gone from $350 a kilo to this, 58 to 63. And that 2,4-difluorobenzylamine went from $550 a kilo to about $125. Now, the acetoacetate has been stable at about $40 to $46, but people have used cheaper alternatives. For instance, LEK in the Czech Republic, I'm really proud because they do chemistry. This is their chemistry, but we originally revealed it to them in discussions through the Clinton Foundation several years ago. They use ethyl acetoacetate as a substitute. Instead of $40 a kilo, it's $2 a kilo. Okay, but it doesn't give you real numbers, but it gives you a feel for how the raw materials have decreased as a cost component of API production. Now let's get into the business of looking at the data in import-export transactions. We all know China and India are huge trading partners for intermediates and raw materials and APIs. Desano supplies really large amounts of dolutegravir to Cipla and to McLeod's in India. India exports dolutegravir API to other countries. Now, in the data that I have, I cannot view the export of dolutegravir API from China to the Philippines or to Brazil, for instance. But I can see India exports to other countries. Look, we don't know the true yield of everybody's synthesis. Cipla's synthesis is a little bit different from Desano's and Emcure's and Aurobindo's and Amneal's and Lonzeal's and so forth. But what we're going to look at is historical price decreases. And what I want to mention here is there's a huge driving force to tell the truth for what the declared value is of an API when it moves in commerce, because the penalties are very large. You can be forced to decrease the cost of your drug, or the penalties can be in the millions of dollars. Just a few years ago, Pfizer was socked with a $3 million penalty for misdeclaring the cost of what one of their shipping Lyrica API from one country into Ireland. Okay, here, let's look at that history. So I confess to you right now, it's tough for me, right? Because as a lecturer, I'm used to people asking me questions, and I'm sorry. Forgive me that I don't know Portuguese. I realize that there's a simultaneous translation going on, and it's difficult. And I hope I'm not going too quickly for everyone. But here, let's look now at a historical bit of movement for how these APIs, this gives us an opportunity. We're watching the declared price of API going from China to India, and we're watching it decrease with time. So look, here we are, February 2020, and Desano is shipping this to Cipla. And look, the API is costing $760 a kilo. And then as time keeps on going on, look, February, June, it starts decreasing. July, November, it goes down. It starts to go down to 720. And these are the kilos that are being shipped. In November through February of 21, it keeps on going down modestly to 690. Then if we look throughout, in March of 2021, there's this giant step decrease from 690 to $495 a kilo. $200 a kilo decrease. And all of these numbers are much less than $4,000 a kilo. And look at the shipment sizes. Now, I can guarantee you that these prices are not solely dependent upon volume. They're also dependent upon what we're watching here is improvements in the synthesis, in the yield, the throughput, the reaction time. So the raw materials input and the overhead and labor input is decreasing. As we go through 2021, here we see another teasing thing. In August, September, another improvement is seen and the price goes down to $416. But then it jumps back up in December. Then at the end of 21, right? November 21 through March of 22, we start seeing these other shipments transition. This improvement is captured and the API price goes down to $395 a kilo. Then we get into the December of 22 and January 23, another giant decrease to 250. And at the end of 23, this is down to 220 to 225 a kilo. This is the history of Desano's shipments to Cipla. Desano's shipments to McLeod's, they have a very similar history. You notice that at the end of 2023, these have gradually or stepwise decreased down to 215 a kilo. This tells us a huge amount, doesn't it? About process improvement, about manufacturing, the impact of volume demand. But it also tells us that a great price for this API would be somewhere in this range, between 215 and 250 a kilo. Oh my goodness, I see a lot in the chat box. I'm going to open the chat box. Oh, forgive me. I can't actually. Oh, people are asking Eloan questions. I wish I could read the questions, but yeah. All right. Here's a summary of what we've been looking at. So we have a little bit of the approximate dates and how we can trace the volume demand and the price decreases. Well, that tells us something, all right, but it doesn't tell us everything. It tells us the API price from China to India. That's a real special relationship, isn't it? We need to know more than that. We need to know who's exporting API to other countries to try and see what an API might cost and what's the driving force to be making your own. Well, here are exports from India. These are selected. I couldn't summarize anywhere near all of them. From India to just South Africa and to Thailand. South Africa has got its own HIV program outside of the Global Fund and outside of USAID, just like Brazil does. And you can see, though, one thing that's worth mentioning is there are a lot of commercial relationships between both Chinese and Indian companies and South Africa. Aspen is a big manufacturer of finished dosage forms. Oh, Eloan, I see that you've raised your hand. Please go ahead. I'll wait for your question.
I have a question, but I'm going to ask in Portuguese. Joseph, o dolutegravir, com esta queda de preço, é de empresas que estão fazendo suas próprias sínteses ou elas são licenciadas pela Vive GSK?
Okay. And I'm waiting, of course, for the translators to think about this, and they can convey it to me so that I'll understand it. Forgive me, I can see that someone was talking.
I'm going to ask in English, okay?
Okay.
Okay. Fortunak, all these companies are producing their own dolutegravir, or are they companies licensed by the GSK or Vive? My question fundamental is that how they can get this kind of price, because if I look for the dose for one tablet, the price from $2,050 is 0.01 cents of dollar. That means that in one tablet you have the price of the API is 0.01 cents of dollar. I did the cost here.
Okay,
but my doubt is, are they licensed companies or are they producing their own APIs?
They're producing their own APIs. So if I'm doing some rough math in my brain, at $200 a kilo, $250 a kilo, it's 25 cents per gram. And that's that's right about 1 cent per tablet.
Yes. Or less, 0.01 maybe. But my doubt is, but dolutegravir is not patented around the world or not?
Okay. This is a special example because of the licenses. So these companies have a license from the medicines patent pool. They did not accept the technology transfer package. They have their own routes of synthesis. So they make the API with their route. So we can look at their patents and we see their synthesis.
Great. Then they are licensed, however, they produce their own APIs.
That's right.
Okay. Now I understood, because I was in doubt about it. It's like maybe they pay something about royalties to the owner or not. Maybe, yes.
Oh, they may pay a royalty of up to 5% to Vive, but that's it, no higher.
Okay. It's fantastic, incredible, fantastic.
Yeah, let's do this in Brazil. Much. Can we do this in Brazil?
Well, exactly what we are going to discuss, because in Brazil, who is supplying is the owner of the patents, supplying with API and others. The price is so high. It's like you don't have the you know, the technology transfer here, the contract accepts the API from the owner, from the Jessica or Gilead. They don't produce their one way, you know.
Oh my.
I
Oh
don't know why.
my.
I don't know why, because I was far from R&Vs, but there's this good discussion with the people participating in this event, in this course now.
Okay. I had a conversation with someone who owns a large pharma company in India and who buys from Desano. And he said, look, we have our process, and that process gets used at Desano. And the other Indian company has a slightly different process, and that process is used at Desano. So that's why our prices are slightly different in the import-export data. But China is there, and China has access to the raw materials, and they're making large amounts. But they aren't selling the finished product. They don't have a license for that. But we have the license for the finished product, and we make the API however our chemists want to do it. GSK and ViiV, they don't care about the price of the API because their price per tablet is more like, I don't know, $100 or $50. So what do they care about the price of the API? So let's look, though. Let's continue looking at the import-export data. South Africa is a country that historically has had really low prices for API from India because they have those relationships. So India will actually ship their API to South Africa, and it'll be made into finished product, and it will be sold under the Cipla brand, or it'll be sold under Ranbaxy's brand. Well, let's look at Loris, okay? Let's look at that really good generic company sending to Aspen. And Aspen does an awful lot of HIV. You see the same sort of a trend of the timing and the amounts increasing and the price decreasing of the API, but it never quite gets down as far as India. India's charging a margin here. So this is probably API that is made by the Indian company and then shipped to South Africa. Thailand is another, it's a contrasting example. So to me, my understanding is that Thailand it's got a very strong, popular HIV treatment program, and they are not as focused on the pricing of APIs to drive down the absolute cost of treatment, and their volume demand is quite a bit smaller than it is total for South Africa. And so their API costs have remained reasonably high. Now, there isn't a lot of data for dolutegravir API exports to Brazil, but here's what I have. So Chromo has supplied to Blonver, and McLeod's has supplied to Blonver, and here are the dates with the amounts and the API pricing. This is a pretty old number, January 23, and it's only 175 kilos, but this is an awfully high API price for versus today. It should be much less. I think I see someone talking, but oh, no, I think I'm okay. I thought there was a question.
I'm sorry, can you hear me?
Yes, I can, Clara.
Yeah, sorry to interrupt, but could you please click on interpretation and select English audio? So this
Oh.
way, if other participants want to ask questions in Portuguese, you'll be able to understand them. I'm so sorry.
Clara, you gotta guide me. Where do I
Okay.
click on participation?
At the bottom of the Zoom, do
Okay.
you see besides share screen on the right of share screen, do you see a button that
the
says
right.
interpretation?
I
At the bottom of
don't.
the screen,
Okay.
there's the share screen button. Can you see that?
No, forgive me. My share screen is on the top, and I don't see a round button to the right of it. Let me click on more, and I'll see if I can. Oh, there's interpretation.
Yeah, so
Okay.
if you can click that and then choose English.
I did. I just did.
Okay.
Thank you.
Thank you so much. I'm so sorry.
No, I really appreciate the assistance. Okay. All right. I'm going to assume that there's no question right now, and I'm going to go on then. So this this is the only data that I have on exports of the API to Brazil, but it's suggesting that it would be well within Brazil's capabilities to be either making your own API at a much lower price, or to be negotiating an API supply from another supplier at a much lower price than you're paying now. That's been a pretty detailed discussion about dolutegravir. So that's as much as I can say. And let's start going on to the other APIs. And I've started here with efavirenz. And it's because dolutegravir has replaced efavirenz as a backbone, right? So efavirenz used to be it's a 600 milligram daily dose, or in some countries, it's 400 milligrams. It used to be in huge demand. The demand maximized at 4,000 metric tons a year when it was in approximately 19 million people. But a good decision was made. We really need to try and decrease the cost of therapy, and going from 600 milligrams to 50 milligrams of dolutegravir as an integrase inhibitor, it's a good deal. So because of that, the demand for efavirenz has gone down quite a bit, but the price has still stayed pretty low. And it's a good example also of what happens for huge decreases in API over time as the process evolves. So we saw the route of synthesis. We saw those molecules on earlier slides. Efavirenz has been through 3 different generations of the routes of synthesis. The first one was 6 steps. Its overall yield from the raw materials was 40%. And it kind of minimized at an API cost of goods, COGS, of around $525 a kilo. And if you think about a person at 600 milligrams a day, a person takes over 200 grams a year of API in their treatment. So that's over $100 a year of API, even before it's formulated. Now, the second generation route of synthesis reduced the COGS down to a minimum of about $225 a kilo. But the third generation is better yet. After you remove all of the protections and deprotections, and if you switch over to phosgene and various different things, and we'll see more about this on the next overhead, but the best cost of goods for the API came down all the way to $78 a kilo. Now, I'm going to compare that, though. Even at $78 a kilo, if a person takes 219 grams a year, that's about $18 to $20 of API as a contribution to the cost of ART. So that makes it just about impossible to get annual costs for treatment down to, say, $50 a person. Now, Loris has that patent on the third generation synthesis. It's listed right here. For a while, Loris supplied over 85% of the API volume because they had this price advantage of owning the third generation synthesis. So they sold API to all of the other Indian companies as well. They sold it to Cipla and Mylan and Hetero and McLeod's. Well, I'll talk a little bit more about, to illustrate again, that impact of what happens of increasing not just the process efficiency, but decreasing the cost of the raw materials. And historically, this has been tracked over several years from, say, 2005 through 2018. So this is the key starting material 4A. This decreased from 120 to 19, whoops. I'm sorry, this number here on the left-hand side is wrong. This is $1,350 a kilo for cyclopropylacetylene. And that came all the way down. Oh, I'm sorry. No, forgive me, please. I have made a mistake. I'm looking at the wrong raw material. This is 9A down here on the right-hand side. When the drug was launched, this could be purchased or manufactured for $120 a kilo. Now it can be bought for $19 a kilo. So its contribution to the COGS decreased from 195 a kilo to 20. Now the cyclopropylacetylene, when the drug was launched, it cost $350 a kilo, and you needed almost a kilo of it to make a kilo of drug. As demand increased, the price came down to $55 a kilo, and now you only need 0.3 kilos to make a kilo of API, so its cost contribution is 15. Similarly, you can see the raw material price reduction in 5A. This isn't incorporated into the drug, it's a catalyst. It's gone here, and its contribution to the COGS went from $220 to $9 a kilo. So it's an illustration. Now, let's look at the import-export data for the pricing of the API. So what we're thinking about now is that when the demand was over 3,000 metric tons a year, the API was $78 a kilo. You'll see it hasn't increased much. Loris is supplying it to Aspen in South Africa at $83 to $90 a kilo throughout 2022. And the volume is substantial, but it's nowhere near 1 million kilos. And Mylan to South Pharma in Thailand, the price there is still pretty low. It's less than $90 a kilo. So a great price for the efavirenz API is somewhere in this range. Now that number is actually lower than the number for dolutegravir, But of course, this dose is 600 milligrams a day. So as a contribution to total antiretroviral therapy, it's much more than the cost of dolutegravir. Tenofovir is another one we really want to get to. Boy, this has been a major component of ART since 2009, and current demand is still over 1,500 tons per year. And here is the fumaric acid pictured. So this is the fumarate salt. This is the route of synthesis. So this is adenine. It's a key starting material. This 4 is called R-propylene carbonate. Huahai in China, very early on, found an enzymatic way of making R-propylene glycol and making this carbonate. And so this can be made for about $5 a kilo. This is 6 is another key starting material. And so is 8. Now I've put a red sort of circle around compound 2. 2 is a crystalline hydrated form known by the acronym of PMPA. And one thing that has changed in the regulatory guidance for this document, for this drug, is that PMPA is now a GMP starting material so that these steps do not need to be run under GMP. TDF has been made by the same bond disconnections ever since it first went into clinical studies. Some of the reagents are a little different. The demand has gone way up, but the yield of this overall synthesis has gone from probably about 3% to over 50%. So here are the key starting materials, and here are the prices in import-export data for these key starting materials. Our propylene carbonate is $12 a kilo. That's probably about 50% margin. So it's a pretty good, that's a pretty good revenue generator for Huahai. DESMP is 15. Chloromethyl isopropyl carbonate is 12. And adenine has been up and down in price. Global demand and various interruptions in manufacturing and COVID have caused adenine to go from $26 a kilo. It went down to 20, then it went back up to 46, and now it's down to 18. It's made in China from carbon monoxide and urea. And PMPA, this GMP starting material, you can purchase this from China at $46 to $55 a kilo. That's an incredibly good price. And I'll emphasize why in another just a second. There are a lot of companies that make TDF API. The Indian companies work closely together. They help each other. If one company runs short of API, they'll buy it from another one, and they won't gouge them on price. One company will buy one API from another con, and they'll sell them a different one to make up for this. There's a lot of cooperativity in order to maximize the efforts of what they do. The import-export data. I'm sorry, I'm going to go back to this because I didn't see what I thought I had. The reason this is such a great price for the starting material, the molecular weight going from 2 to the API doubles. So if you start with 1 kilo of 2 and you do the manufacturing process, you get about 1.5 kilos of API. So this only contributes about $35 a kilo to the API price. That's why it's so efficient as a starting material. Here's a summary of the import-export data for TDF. And a great price looks to be less than about $100 a kilo. Well, this is $300, 300 milligrams per day. So $100 a kilo would be about $11 a year as the API contribution to therapy. China sells it to India at this price, $78 to $81. India sells it to Pakistan. India provides it to Brazil at about 136 to 140 a kilo. India sells to Colombia and Vietnam at a somewhat lower price. India also provides Egypt, Thailand, and some to Vietnam at a price that's pretty comparable to Brazil's price. And to companies that are using it for their high-income country manufacturing, Ireland, Canada, the US, they sell it at a much higher price, as you can see. Well, I wanted to go into a little bit of detail about the API pricing because it illustrates a few things that I've talked about. This is mapping the impurity profile through process development. This is what is not contained in a patent. It's contained in a product development report that is part of a drug master file. So this describes a history of how a company modifies and optimizes the process to make the API as pure as they can, and how they decided the quality requirements. So this is an HPLC of the drug substance and the impurities that are present. Here's a process flow chart. It tells you what you do in each step of this stage of a synthesis. This is pulled out of this publication. This is from, I think this is from Organic Process R&D Journal. And it shows stage 2 of the tenofovir synthesis. So it shows where you have the reaction going on and the workup and the flow streams of all of the material and what goes into each stream when you're making the product. And here's another publication about optimizing typical approach to process improvements. I'm doing it for a certain reason. I'm highlighting here a few points to make. One of them is that making tenofovir, it was a really interesting approach, but in making tenofovir, you only use a very small amount of solvent, so you have a very high throughput. So there's only 4 kilos per kilo of the solvent N-methylpyrrolidone that goes into the reactor vessel, so that in a 4,000 liter vessel, you can generally you can make up to 900 kilos per batch of the API. That's a huge batch. So a company that's making 300 kilos per batch is at a large disadvantage to another company that has a batch size of 900 kilos in the same size reactor. Another thing is to illustrate the yields. So this is pretty much the same chemistry, but the yield of each step improves as you understand the process better. So the yield at small scale and the yield that's in the patent application is about 20%. When the Indian manufacturers first went generic, the yields improved to about 36 to 44%. But very rapidly, it was understood that most of the product was being lost. It was being destroyed during the isolation and workup of the reaction. Because if you took an HPLC analytically, you could see that a much higher yield was originally present. Now, I won't read to you these issues. So the problem is this, it's right here. Let's look right there. Huge losses during the workup and the isolation. These are the reasons why, but I won't go into them. The solution was discovered by serendipity by a student of mine. Her name is Frajavan Tali. This is way back before 2010, and I'm going to illustrate it by showing you part of the process development. Okay, here's what people do in process development. This is a chromatogram. You're looking at a reaction, and you're looking at the conversion of the starting material over here to the product over here. But this is almost like watching television of a reaction, because you see a peak for each of the impurities that's generated during the reaction. And you can see that if you look at the area percent for starting material, byproducts, and impurities, when the reaction is optimized, when it's finished and it reaches a maximum, there's only 75 area percent for the desired product. That's terrible. And there's a lot of really small impurities. Well, this is what made it so difficult. And this is from a 300 kilo batch size. But that's what makes it so difficult to optimize the reaction, because the workup and isolation was so tedious. These are the structures of the impurities. And as Dr. Pinheiro, my hero, has mentioned, you need to know the impurities that are present in a process and the degradants during a formulation in order to be able to understand how to make a process work. Here's where serendipity came in. A student was running this reaction for the first time. She ran that reaction as it's described for a 300 kilo batch, and she was only doing 30 grams. And she made a mistake in the reaction workup. When the reaction was finished, she did not add water. She added isopropyl acetate first. And she came into me and she showed me the reaction flask, and she said, "Dr. Fortunak, I have a problem. I have this white crystalline material that's coming out of the reaction solution. And I took an HPLC of the solution, and there doesn't seem to be very much product in there." And lo and behold, she was isolating a crystalline solvate that was very pure of the desired product, and all of the impurities got left behind. And there was no aqueous workup, there was no back extraction, there was no distillation, there were no yield losses, no decomposition. She got an isolated yield. Her yield in solution was 81%, and she isolated 77% of it. And its purity was right here. It was greater than 99.7% pure. This is actually fumaric acid, so this is not an impurity. And this is a picture of the student. She's a dentist. She works in North Carolina now. She has her own dental practice. And while she was pursuing her master's degree in chemistry, she was also a cheerleader for the Washington professional football team. Here's tenofovir alafenamide. Now, tenofovir alafenamide, it was so exciting because it was better science. It was a better prodrug. This was the only thing that was in circulating blood plasma, and it was only after the drug got into an HIV-infected cell that an enzyme would reveal the active drug. So this different prodrug reduced the dose from 300 milligrams a day to 25. Well, it went through development, it was approved, and it's used in some countries. But the demand is low. It was licensed by Gilead to the medicines patent pool. People took up the licenses, but nobody wants the drug. Well, there are three problems, actually. One is that it's not actually better than TDF for suppressing the HIV virus. Secondly is it might be more toxic than TDF. And thirdly is, even though it's a lower dose, it's more expensive because the API is so much more expensive to manufacture. It is more than 15 times as expensive as TDF. Okay, look, making TDF and TAF, it's the same starting material. I'll remind you again, you can buy it from China for this much as a GMP starting material. 1 kilogram of 2 gives us about 1.5 kilograms of TDF. So TDF probably costs about $60 a kilo to manufacture, maybe even a little bit less. But TAF is much more complicated to produce from 2. This is the route of synthesis for TAF. This is what's published. So you need to make this piece, then you need to couple this, you need to couple the starting material 2 with phenol to make 325. Then you couple it with this, and you get a mixture of diastereomers at phosphorus. You need to separate those diastereomers. The literature says that you separate it by simulated moving bed chromatography, but that's not the way that it works. There's a process where companies, they purify to remove the unwanted diastereomer by crystallization to get Puritan off of aralfenamide. But the problem is, with that low of a dose and with that low of a demand, the API is really expensive. Here is a more recent patent that doesn't belong to Gilead of another way of crystallizing this mixture cleanly. Here's the import-export data for TAF. Look at the demand. It's not very big. Okay, multiply this by 12 in order to get the equivalent amount of TDF, and it looks better, it's closer. But look at this, is over the course of an entire year. So Sidmax exported 65 kilos in 4 shipments to Argentina. The lowest price is over $2,000 a kilo. Compare that to the price of TDF, about 100. Same thing here. The problem with TAF is that it's, until more recently, there's no indication that it can be cost competitive with TDF as an API. And it's not clear what a great price would be for TAF. I would suggest that if you have somebody who's really ambitious, you might be able to solve this problem. And putting TAF instead of TDF, it could be really good. Okay, nevirapine is an old drug. Nevirapine used to be in demand in over 1,000 metric tons a year. This is its route of synthesis. There are only 1 or 2 companies in India who make the API, and Desano still makes it. This route of synthesis, it's got modifications to it. A great price for nevirapine would be somewhere in this range, $110 to $160 a kilo. Its dose is 400 milligrams a day. So you divide this by 7 to get the cost per year of the API. You notice the volumes shipped are just not that high. This is over almost 3 years. So Desano still sells it. They supply it to Cipla and to McLeod's. Hetero sells it to Thailand at this price. There's still some demand in South Africa, and there's a little bit of demand in other countries. Here's atazanavir. Atazanavir is just a fantastic drug from a chemistry perspective because it was such a good synthesis. But it's for second line or some small pediatric indications. The volume demand isn't very high. It's a 300 milligram a day dose, just like TDF. This is part of the route of synthesis. This is one starting material. This is called the Bach epoxide. And this biaryl hydrazine is another key starting material. You put these two pieces together, then you remove these Bach groups in a stage 3, and you react it with something called MOC-TERT-leucine, or MOC-T-LEU. This is methoxycarbonyl tertiary leucine. So the synthesis is pretty short. The processing input is not that high. The cost of the API is largely dependent upon demand and the pricing of these 3 key starting materials. The Bach epoxide comes from China at about 127 a kilo. MOC-T-LEU is 55 a kilo. And the biaryl hydrazine, it's hard to get a good price on this. It seems to be around $90 a kilo, but this may be going up. Here's the import-export data. So you can see that through 2023, about 3 tons went to Colombia. So Colombia wants atazanavir and uses it heavily. Cyprus is where most of the demand for Europe goes, 750 kilos. There's a modest amount that goes to Russia for second line antiretroviral treatment. And you can see that the price to a high income country, Canada, automatically it goes up. Cyprus is actually able to distribute into Europe and charge kind of European prices from granules, so their price is higher as well. And here is from Loris to Pliva. Pliva in Germany, not in the Czech Republic, at 935 a kilo. A great price for atazanavir API would be somewhere in here. Now let's get to the nucleosides. So lamivudine still has a really high demand, over 1,000 metric tons a year. Lamivudine and emtricitabine, they differ only in that emtricitabine has a fluorine right here. So they have very similar routes of synthesis. You start from, you have to have a chiral ligand, and this is menthol. Menthol is very inexpensive. You make a sugar analog. So this is a thio sugar. You activate it and do something called a Vorbruggen coupling. And this is called the coupled lamivudine ester. We're going to see the coupled emtricitabine ester as well. These are common GMP starting materials now, and they can be very efficiently turned into the API. This was the manufacturing route at GSK. The patents have expired. Most companies do a synthesis that is very close to this one. although they might use a different chiral alcohol. Menthol costs about $11 to $12 a kilo. Cytosine is the nucleoside. It costs $10.40 to $14 a kilo. Compound 11. So that's this. It can be purchased for about $21 a kilo, but 13 is the real big deal. It goes as low as $27 a kilo. So because of this, lamivudine, a great price for lamivudine is less than $100 a kilo, $73 to $88 a kilo. Look at the demand. The shipment through 1 year from China to India is 425 tons. From India to Brazil, from Aurobindo, it's quite a bit more expensive. Here's the pricing from other countries. Thailand and South Africa get pretty good pricing, actually very good pricing, and Argentina is sort of an intermediate price. India sells it to GSK in Singapore for their use, right? So an originator company is buying the API from a generic at $320 a kilo. And here's emtricitabine. So the demand is a lot less. You know, if I had HIV, I might prefer to take emtricitabine, but there's no real evidence that emtricitabine ART is superior to that of using 3-TC. But there's a big difference in the API pricing. There's a difference in dose. 3-TC is 300 milligrams a day and FTC is 200. Cytosine is $11 to $14 a kilo, but the 5-fluorocytosine costs from $34 to $56. Generally, you see it for about $45 a kilo. So that's why the price of the API for emtricitabine is quite a bit higher than for lamivudine. This shows there are dozens of different routes of synthesis that all look quite similar. They're each a little bit different. And it also shows, this is a recent publication. This comes from Paul Watts in South Africa. And I'm pretty sure that other generic companies have already done this kind of chemistry. Anyway, look, there are multiple routes of synthesis for both lamivudine and emtricitabine. These are the prices of the key starting materials. The coupled ester, right? The coupled ester for lamivudine was about $27 a kilo, but it's almost twice that much for emtricitabine. So a great API price for emtricitabine is $180 to $240 a kilo. So it's more than twice that of lamivudine. So here we are. And I'm going to finish up by showing you just a couple of overheads. I want to show you what a real manufacturing filing looks like in order to contrast that with what we saw in a typical process description in a patent. This comes from an actual drug master file. So here's the table of contents. Here is actually some descriptions of what's going on. And this is a little bit of it shows you part of the table of contents showing the process development report. Look, the rationale for the designation of the starting materials. Please remember, when you start good manufacturing practice, the expense increases, it just about doubles, because good manufacturing practice is expensive. So the later in the synthesis you can start GMP, the better off you are. Here is the actual route of synthesis, and I've whited out some of these. I blocked them out because the person who supplied this to me said, you can show this, but please don't show this. That would be confidential information. Here is what would be part of the closed section of a drug master file. It shows a table of what goes into the master batch record for one step in the API production record. It shows the input and the output and the batch size. Not a very big batch. This is the process flow diagram that shows what goes into and what comes out of each step in the processing of that stage of the synthesis. And this is the actual process description. So the actual amounts come from the real batch description. So you look at a table in the batch record, and it tells you exactly how many kilos of potassium carbonate, hydrochloric acid, sodium bicarbonate, water, all of this other stuff goes into the synthesis. And those inputs are described here. So it says water and potassium carbonate. So you would go to the table to see how much, then you stir it. Then you add the amounts of starting materials 1 and 3, and it's charged while maintaining the temperature at 30 degrees centigrade plus or minus 5 degrees. After that, you raise the temperature. You maintain it for approximately 10 hours. You take a sample, you test it for remaining starting material by HPLC. It should be less than 2%. If it doesn't comply, what you do is you continue it and you sample it again every 3 hours. And so what I'm doing this for is to convey to you that a real process description has much more detail than what goes into a patent. And it tells you how much you get out of this. It tells you the actual conditions. So for instance, it says the temperature of the reaction is increased but kept below 55. The toluene is removed via vacuum distillation at a pressure of greater than 650 millimeters of mercury until no more distillate is observed. Then you add more toluene and you distill it again. It's an azeotrope to get a water limit of not more than 2.2%. To those of you who've done chemistry in the audience, this is a telling illustration of the difference between what a real process description is that you need and what's in a patent. So here's a summary for us, okay? If you have leapfrogging technologies, you can put them in place and you can do better than what other people have done. Generic companies, especially in China, they do that. Here's an argument for national and regional independence and self-determination, but you know that even better than I do. But here are a few final points. Patents keep drug prices very high, and they create barriers to API production. But for the generic people who are able to make the API, they stimulate improving the production. They stimulate innovation. Generics usually have their own process, not the originator's API process. even for really old drugs, the generics have their own manufacturing plant. They adapt a process to their needs. When you sell APIs, typical margins are about 20%, maybe 30% at the most. If there's a lot of competition like TDF, it may only be 10%. These are the cost components. The reason that a lot of HIV APIs are low price is because the capital investment has been fully paid off. It's been amortized. Now, the pricing that we see in the import-export data, it doesn't include shipping costs or tariffs. And to finalize, hey, patents don't describe a manufacturing process. And this is, I believe, that's 2 more overheads. Here's a table that summarizes what I put in as my take on from the import-export data of what's great API pricing. Here's the dose. I didn't finish this table, so forgive me. I'm going to leave it at this. But if I had import-export data for Brazil, I included it in this column. And in summary, China supplies huge volumes of a lot of the HIV APIs to India. India produces the same drugs. Cipla buys dolutegravir from Desano, but they also make their own. Dolutegravir, tenofovir, lamivudine, they're really large volume APIs. And this really is just, it's a summary and I'm finished. Thank you for giving me so much time to discuss with you. I'm really grateful to be here with you. I'd love to continue the dialogue, and I'll see you again tomorrow when we listen to my hero, Eloan. So I am open for questions.
Muitíssimo obrigada, professor Joseph Fortunak. Foi um grande aprendizado. Espero que você esteja conseguindo me escutar. Caso sim.
I can, thank you.
Ótimo. Hoje estamos já encerrando o horário. Creio que não é possível abrir para grandes perguntas, mas temos aqui entre nós farmacêuticos, químicos, que eu tenho certeza que entenderam muito mais sua apresentação do que eu, que sou apenas uma advogada, mas de todo jeito, excelente. Por favor, quem quiser se manifestar agora para alguma questão, amanhã o professor estará novamente conosco, então agradeço de novo a gentileza por compartilhar tantos conhecimentos e informações. Caso alguém queira se manifestar agora ou não, acho que amanhã a gente pode iniciar a nossa conversa abrindo para perguntas. Gabi, você gostaria de fazer alguma questão?
I realize it can be difficult to ask questions, especially after a long presentation. And while we're waiting, look, I just have to express something. You know what? I really adore Eloan. She actually shaped my life. It is always a privilege to be with her.
Professor, muito obrigada pelas palavras a Eloan. Eloan é uma pessoa muito querida aqui por todos nós. As suas palavras sempre da Eloan e a sua agora elogiando, ela nos comovem bastante.
Fortunak, the pleasure is absolutely mine. Because you are big friends for a long time, since Jet Car, in 98th Street, exactly. The crazy woman that somebody present to you. Yes, since that time, I fight against patents, I fight against high price and so on. However, we will be together tomorrow, and I'm so happy with your presentation. So happy. And tomorrow we'll continue to discuss a bit, all right? About now I'm going to take your API's price and put in my spreadsheet for final formulation and show everybody how the final formulation is built and the price comes out. All right?
That's beautiful. I love you, my dear.
Me too, me too.
Bom, gente, muito obrigada a todos pela presença. É bom tê-los aqui. E estou encerrando agora esse webinar e amanhã a gente continua às 2 horas e seguimos mais uma vez pontualmente até 5h30. Muito obrigada mais uma vez, professor. Muito obrigada, Eloan. Nos vemos amanhã.